development of most multicellular organisms, a single cell gives rise to many different types of cells, each with a different structure and corresponding function. Essentially, a fertilized egg becomes a different organism over time. So the fertilized egg gives rise to a large number of cells through mitotic cell division, but the process of cell division alone can only lead to increasing numbers of identical cells. So as cell division proceeds, the cells not only increase in number, but also undergo differentiation, which is a process in which a cell becomes specialized in both form and function. Stem cells are undifferentiated cells that have two important characteristics. They are unspecialized, and they are capable of renewing themselves by cell division. So under certain natural or experimental conditions, they have the ability to differentiate into one or more types of specialized cell. The various types of cells arrange into tissues, which are organized into organs and ultimately into organ systems and organisms. So stem cells are the cells that you start with, and they become all of the cells that you end with. That process of differentiation is extremely important. Nearly all of the cells of a multicellular organism have exactly the same chromosomes and DNA, and so during the process of differentiation, only specific parts of the DNA are activated. The parts of the DNA that are activated determine the function and specialized structure of a cell. Because all cells contain the same DNA, all cells initially have the potential to become any type of cell. But once a cell differentiates, this process cannot be reversed. To really understand stem cells, you first need to understand the levels of potency. So potency specifies the differentiation potential, or the potential to differentiate into different types of cell of the stem cell. The totipotent or the omnipotent stem cells can differentiate into embryonic and extraembryonic cell types. They can become anything. So such cells construct a complete viable organism. These are produced from the fusion of an egg and a sperm cell, and they only stay totipotent for the first couple of cell divisions after fertilization. Pluripotent stem cells are the descendants of those totipotent stem cells, and they can give rise to all the cell types that make up the body. Embryonic stem cells are considered pluripotent. IPS, or induced pluripotent cells, are made from adult cells chemically in the lab, so we can make cells become pluripotent again, but not totipotent. Multipotent stem cells can differentiate into a number of cell types, but only those of a closely related family of cells. So adult stem cells and cord blood stem cells are all multipotent. Oligopotent stem cells can only differentiate into a few things. In your lymphatic system, you have stem cells that can turn into both the different types of B and the different types of T cells, but nothing else. So oligopotency is very limited multipotency. Unipotent cells are cells that can produce only one cell type. They still have the property of self-renewal, which distinguishes them from non-stem cells. Say, for example, progenitor cells or muscle stem cells. They can become more of the same thing. Plant stem cells are innately undifferentiated cells located in the meristems of a plant. Plant stem cells serve as the origin of plant vitality, as they maintain themselves while providing a steady supply of precursor cells to form differentiated tissues and organs in plants. Two distinct areas of stem cells are recognized in plants: the apical meristems at the tips and the lateral meristems at the branches. Plant stem cells are characterized by two distinctive properties. They can create all differentiated cell types, and they have the ability to self-renew such that the number of stem cells is always maintained. Plant stem cells never undergo aging process, but immortality gives rise to new specialized and unspecialized cells, and they have the potential to grow into any organ, tissue, or cell in the body. Thus, plant stem cells are totipotent cells equipped with regenerative powers that facilitate plant growth and production of new organs through lifetime. Unlike animals, plants are immobile. As plants cannot escape from danger by taking motion, they need a special mechanism to withstand various and sometimes unforeseen environmental stress. Here, the meristems is what empowers them to withstand harsh external influence and preserve life. There are two main types of animal stem cells. You have the embryonic stem cells, which are made from or derived from a 5-day-old embryo. They are pluripotent, not totipotent, and they have the capacity for long-term renewal. Adult stem cells are undifferentiated cells found in certain organs and differentiated tissues. So they are somatic stem cells. They maintain and repair the tissue in which they are found. But please don't think that they only occur in adults, they also occur in children as well. In the 3 to 5 day old animal embryos, stem cells give rise to the entire body of the organism, including all of the many specialized cell types and organs, such as the heart, lungs, skin, sperm, eggs, and all other tissues. In some adult animal tissues, such as bone marrow, you have the hematopoietic stem cells. Groups of stem cells generate replacements for cells such that are lost through normal wear and tear, injury, or disease. I mean, think about the red blood cell. Red blood cells don't have a nucleus. They're not capable of mitosis. So the only way that you're able to produce new blood is by using your hematopoietic stem cells to do so. And that's in the red bone marrow, in the core of most of your bones. Stem cells are being used to study normal growth, development, and differentiation. If we can really understand stem cells, we can use them to repair a wide variety of diseases. Cell-based regenerative therapies are treatments in which stem cells are induced to differentiate to repair damaged or destroyed tissues, say for example to regrow an organ or a limb, or repair damage to the spine after an accident. So there's definitely a lot of potential there. Currently, we're using stem cells to screen new medicines to check for safety of medicines for human use. But stem cells offer a renewable source of replacement cells and tissues for a person. Bone tissue from bone marrow cells, cells from the pancreas to produce insulin to treat diabetes, all sorts of different things that we could use stem cell for in the future. There are many pros and cons for the type of stem cells that you use. Embryonic stem cells can become all of the types of the cell in the body, so they're pluripotent, whereas adult stem cells are only multipotent, so they're maybe less useful. Embryonic stem cells are very easy to grow, and they can divide indefinitely. But adult stem cells can be hard to isolate and sometimes can be very difficult to grow in culture. However, embryonic stem cells, we don't really know how they're going to work with the immune system. But if you culture, say, a new pancreas from somebody using cells from their pancreas, it's very, very unlikely that the body would be able to tell it apart because it's not apart. It's made from the cells of that person. So it's very unlikely to be rejected. So there's definitely some major possibilities for adult stem cells in organ generation.